Good morning, everyone. Today I'm going to present a seminar on physiology of sleep. My moderator is Dr. Veena. What is sleep? It is defined as an unconsciousness from which a person can be aroused by sensory or other stimuli. Brainwave frequencies relating to sleep. What is sleep? It is defined as unconsciousness from which a person can be aroused by sensory or other stimuli. Brainwave frequencies related to sleep. First is alpha wave. Alpha wave is 8 to 12 hertz. 12 hertz waves. It is present when the person is relaxed, calm, lucid, or not thinking, or when a person is sitting with eye closed. Then beta wave. Beta wave is a wave with frequency of 12 to 30 hertz. It is present when the patient is awake, normal, alert, or trying to concentrate on. Then third is theta wave. Theta wave is a wave with frequency of Theta wave is wave with frequency of 4 to 7 hertz, and it is present during deep relaxation and meditation and at the time of NREM sleep. And delta wave is 1 to 4 hertz wave. It is present in slow wave sleep.
So why not order alpha first and then beta? Then it's very difficult to remember. The waves with the highest frequency. So beta, just like you have done in the graph in the right manner. So you have to remember and mention about beta first because beta is with the highest frequency. देखा ना कम हो रही है beta 12 to 30, right? उसके बाद alpha कम हुई 8 to 12, उसके बाद theta कम हुई 4 to 7, then delta कम हुई 1 to 4. So we remember it by D tab, DTAB. Delta सबसे low frequency, theta then. So अगर आप ऐसे alpha एक भी सीधे भी नहीं कर रहे आप तो बीच में theta डाल दिया. So the proper order is you have to remember either by increasing frequency or decreasing frequency. So that is the right manner. So आप वैसे ही learn करें. नहीं तो मतलब आपको बिल्कुल नहीं याद रहेगा कि कौन से. It's very important to remember this. You will recognize when you go ahead in your seminar. So better you mention, write, and remember in the proper manner so that it is easy to do. Confusing हो जाएगा कि कौन सा ज़्यादा. Otherwise, easy will be very difficult to report. You have to remember which is highest. So it is beta पहले बोलो तो then you can turn around the order. That is not a problem. Keep it in order.
Using EEG, electrooculogram and electromyogram recording, it is possible to recognize two states of sleep, and NREM sleep and REM sleep, non-rapid eye movement sleep and rapid eye movement sleep. Rapid eye movement sleep. It is also known as paradoxical sleep. Muscle tone is depressed in this sleep, indicating strong inhibition of spinal muscle control areas. There is burst of rapid eye movement. Brain is highly active. Overall brain metabolism during this sleep is increased by 20%. Dreaming is present. Dreams that are remembered are from an REM sleep. This sleep is important for memory consolidation, and body temperature regulation ceases temporarily, and phenylalanine clitoral tumescence occur. It has two components: phasic RAM sleep and tonic RAM sleep. In phasic RAM sleep is sympathetically driven state characterized by irregular burst of rapid eye movement and muscle twitches. And tonic RAM sleep, in tonic RAM sleep, there is generalized decrease in tone of skeletal muscle, except for extraocular muscles and diaphragm muscles. In tonic RAM sleep, we'll see sawtooth waves and train of triangular serrated 2 to 6 hertz waves. This EEG in tonic RAM sleep is similar to the EEG of N1 stage of RAMS, NREM sleep. The NREM sleep, it is also called as orthodox sleep. Most physiological function, including brain activity, is markedly lower than wakefulness. That is a peaceful state relative to waking. Dreaming occur, but it is abstract, disorganized, unreal, and it is not remembered. So NREM stage of sleep is associated with night terrors, somnambulism, somnology, enuresis, and bruxism. Stages of sleep. Stages of sleep are classified according to two classifications. One was given by Rechensteffen and Kales in 1968. Classified who classified sleep into a stage of wakefulness, then NREM stage 1, NREM stage 2, stage 3, stage 4, and stage 5, and a recent classification given by American Academy of Sleep Medicine, which has classified sleep as stage of wakefulness, then N1 stage, N2 stage, N3 stage, and stage of rapid eye movement sleep. So in wakeful stage or stage W, in EEG, there will be alpha waves, that is waves of 8 to 13 hertz frequency, which are present when the eyes are closed. Eye movement and eye blinking will be present. And on electromyography, high tonic activity and tonic movements will be seen. In stage N1 of sleep, there will be theta waves. These are 4 to 7 hertz waves of 4 to 7 hertz frequency. On electrooculogram, you will see slow eye movements, and on electromyography, there will be a tonic activity the tonic activity will be slightly decreased from wakefulness. Then in stage N2, there will be sleep spindles. Spindles are 12 to 14 hertz burst and K complexes seen. So these are sleep spindles. These are 12 to 14 hertz burst of waves, and these are K complexes, which is a prominent negative wave followed by a prominent positive. In a stage in stage N2 of NREM sleep, on electrooculogram, there will be no movement, and on electromyogram, there will be low tonic activity. In stage N3, there will be high amplitude, that is of more than 75 microvolt slow waves of less than 2 hertz frequency. High amplitude slow waves of less than 2 hertz frequency. On electrooculogram, there will be no movement seen, and on electromyogram, there will be low tonic activity. In RAM stage of sleep, there will be sawtooth waves and theta activity seen. On electrooculogram, there will be rapid eye movement, and on EMG, there will be atonia. This is stage 1 with theta waves of 4 to 7 hertz frequency, stage 2 with sleep spindles and K complexes, and stage 3 is high voltage, high amplitude waves with decreased frequency of less than 2 hertz. Sleep architecture, the sleep starts with NREM sleep, the NREM sleep that lasts for about 80 minutes, which is followed by a RAM stage of 10 minutes. This 90-minute cycle is repeated at least 3 to 6 times each night. In successive cycle, the amount of RAM sleep increase in NREM decrease, as seen in this graph. The blue color shows the RAM sleep. So with successive cycles in night, the amount of RAM sleep increase and NREM decrease. In a healthy adult spend about 5% of the sleep in N1, 50% of the sleep in N2 stage, and 20 to 25% of the sleep each in slow wave sleep, that is NREM 3 plus NREM 4 and RAMs. Physiologic effect on autonomic nervous system. During NREM sleep, there is relative increase in the parasympathetic tone, while in RAM sleep there is brief surge in both sympathetic and parasympathetic tone. On cardiovascular system, during NREM sleep, the blood pressure, heart rate and cardiac output all decreases. Lowest value of cardiac output is seen in slow wave sleep. Slow wave sleep is also called as delta wave sleep, and it is equals to NREM stage 3 plus NREM stage 4. The REM sleep, in REM sleep, the BP, heart rate and cardiac output varies. Sometimes it becomes equals to, or it can become more than wakefulness also. Pulmonary system. In pulmonary system, the minute ventilation falls with the onset of sleep. During NREM sleep, the minute ventilation falls. There is reduction in tidal volume. And the sensitivity of chemoreceptors to O2 and CO2 decrease. Hence, the threshold for CO2 increase, that is high end tidal CO2 is required for respiratory drive. In REM sleep, there is increased frequency of respiration and reduced regularity. The tidal volume is again reduced, but it is reduced further in comparison with that of NREM sleep. There will be lowest level of minute ventilation, and threshold for response to CO2 increases even further. During NREM sleep, the brain and body temperatures are downregulated due to decrease in hypothalamic set point. And during the REM sleep, it is not regulated. There is no effect of on NREM sleep or sexual functions. In REM sleep, there will be penile erection and increased blood flow in the vagina. During NREM sleep, the growth hormone increases. The ACTH rises in the initial part. However, the ACTH rise in the final hours during the REM sleep. There will be increase in insulin release and increase in plasma renin activity. During REM sleep, there will be testosterone release. There will be rise in ACTH that occurs just before awakening. There will be increased insulin. There will be decreased release of insulin. Decrease on plasma renin activity and inhibitory effect on gastric acid secretions. In musculoskeletal system, in REM sleep, there will be 8. Now the reticular activating system. The RAS neurons are the center to the regulation of wakefulness. So these are heterogeneous regions that run through the core of brainstem, and these comprises of a series of well-defined cell groups with identified neurotransmitters. So these systems produce cortical arousal by two pathways. One is dorsal pathway and one is ventral pathway. The dorsal pathway, it passes through the thalamus, and the ventral pathway, it passes through the hypothalamus and the basal forebrain. In the dorsal group, the dorsal pathway, there will be the cholinergic, pedunculopontine, and later dorsal tegmental nuclear projections from the brainstem that relay in the reticular nucleus of the thalamus, and that projects to the cortex via thalamocortical transmissions. And in the ventral route, that passes through the hypothalamus and basal forebrain, that receives projection from locus ceruleus, non-adrenergic projections from locus ceruleus, serotonergic projections from dorsal and median raphe nucleus, dopaminergic projection from ventral tegmental nucleus, periaqueductal gray matter nucleus, histaminergic projection from tuberomammillary nucleus, glutaminergic projections, and cholinergic projection from the basal forebrain. Neurons in each of these sites send axons through the lateral hypothalamus to the basal forebrain and cerebral cortex. Now, the various wake promoting neurochemical system. First is acetylcholine. Acetylcholine is released from the basal forebrain and in brainstem. What is basal forebrain? It is a region that is marked in this picture surrounding the front of hypothalamus. So this basal forebrain contain cholinergic nuclei, which are medial septal nuclei, as seen in this picture, medial septal nuclei, lateral dorsal tegmental nuclei, and medial septal nuclei, dorsal band of Broca, and nucleus basilis of Maynard. These are the three nuclei that are present in basal forebrain that secrete acetylcholine. Now, the nuclei that are present in the brainstem are lateral dorsal tegmental nuclei and pedunculopontine tegmental nuclei. These nuclei secrete acetylcholine. The second neurotransmitter that is involved in wakefulness is norepinephrine. Norepinephrine is secreted from locus coeruleus, and locus coeruleus is an elongated nucleus just beneath the floor of fourth ventricle. Serotonin. Serotonin is released from the dorsal raphe nucleus and other nucleus scattered along the midline of the brainstem. So dorsal raphe nucleus is the main source of serotonin for wakefulness. Then dopamine. Dopamine is released from substantia nigra and ventral tegmental area. Fifth is histamine. Histamine is a major neurotransmitter that is involved in wakefulness. It is secreted from the tuberomammillary nucleus of the hypothalamus. Tuberomammillary nucleus is a small cluster of cells adjacent to the mammillary body that is present in the third ventricle. So the histamine that will act on the wakefulness promoting areas, which are prefrontal cortex, the basal forebrain, the thalamus, the ventrolateral preoptic areas, and the lateral hypothalamus. Now, orexins or hypocretins. Orexins or hypocretin neurons, these are present exclusively in the lateral hypothalamic area, peripharyngeal area, and posterior hypothalamus. These neurons in the hypothalamus, they make two neurotransmitters, orexin A and orexin B. Orexin A that act on the receptors orexin 1 and 2, and orexin B act on orexin B only act on orexin 2 receptors. The orexin neurons project widely. The orexin neuron, they project widely and innervate the arousal region, which the orexin neuron, they particularly innervate all the arousal regions, and particularly the locus coeruleus area and the tuberomammillary nucleus, which release histamine, which is a major neurotransmitter for the awakening. So orexin stabilizes wakefulness by interacting with all the arousal neurotransmitters. Other hormones and peptides that promote wakefulness are cortisol, substance P, neurotensin, hypothalamic peptide, corticotropic releasing hormone, vasoactive intestinal peptide, and thyrotropin releasing hormone. Now, pathway for arousal and sleep for a sleep-wake cycle. So, regulation of a daily sleep-wake cycle is mediated by two opposing drive, that is homeostatic sleep drive, that is S, and circadian sleep drive, that is C. This theory was developed by Alexander Bobley and Fritz. The homeostatic process, that is the S drive. This states that the longer the individual is awake, the greater is the homeostatic drive to sleep. It depends on the accumulation of adenosine, which is further dependent on the time of wakefulness. So there is rise in adenosine level, which is seen in basal forebrain during prolonged wakefulness. Due to this buildup of adenosine, this lead to disinhibition of VLPO, which is a sleep promoting area, release of GABA, GABA in the sleep circuit, and that which facilitate the onset of sleep. Then second is circadian process, that is C, that is mediated by light. When light falls on the retina, it stimulates it travels through the retinohypothalamic tract. Through retinohypothalamic tract, it will stimulate the suprachiasmatic nucleus, which is a pacemaker for the circadian rhythm. So through suprachiasmatic nucleus, it will stimulate supraventricular nucleus, which will stimulate dorsomedial nucleus. Dorsomedial nucleus will release glutamate and TRH, which are stimulatory hormone for sleep, and which will stimulate lateral hypothalamus, and lateral hypothalamus will release orexins, which are the neurotransmitters for maintenance of wakefulness of sleep. Wakefulness. these orexins, they will promote the release of norepinephrine, serotonin, dopamine, and acetylcholine. When the light fades, there will be accumulation of non-epinephrine and serotonin. There will be negative feedback on orexin. So orexin will be reduced. So wakefulness cannot be stabilized. The wakefulness from the sleep promoting areas, VLPO and GABA will take charge, suppress all the arousal neurotransmitters, and sleep will be facilitated, and melatonin will be. So release of melatonin is mediated by this circadian process. Retinohypothalamic fibers that synapse with the suprachiasmatic nucleus. Suprachiasmatic nucleus is involved in the is the pacemaker of the circadian rhythm. So the retinohypothalamic fibers, when they synapse with the suprachiasmatic nucleus, then they are connected to the sympathetic preganglionic neurons in the spinal cord. These sympathetic preganglionic neurons, they project to the superior cervical ganglion, and from the superior cervical ganglion, the postganglionic fibers, they relay in the pineal gland that secrete melatonin. So the suprachiasmatic nucleus is involved indirectly in melatonin release. This cyclic activity of suprachiasmatic nucleus creates a rhythm for melatonin. Now the sleep promoting regions for NREM sleep promoting region, there is preoptic area. Preoptic area is anterior end of hypothalamus, just about the optic chiasma, and it has two parts, that is the ventrolateral preoptic area and the median preoptic area. They act by inhibiting arousal promoting regions through GABA. These areas release GABA and they act by inhibiting arousal promoting regions through GABA and they promote sleep. Similarly, the RAM sleep promoting regions, pons is the major area that promote RAM sleep. The RAM sleep is controlled by cholinergic neurons, which are located in the brainstem. The RAM sleep is promoted by these REM sleep is promoted by these two neurons, that is lateral dorsal tegmental nuclei and pediculopontine tegmental nuclei. The acetylcholine released from these two nuclei will depolarize the thalamic neurons to promote the transmission of information from the thalamus, driving the cortical activation that is required for the complex dream of REM sleep. Now, the atonia that is seen during REM sleep is again mediated by this LDT and PPT neurons. These neurons will release acetylcholine and that will stimulate the neurons in the ventromedial medulla. Ventromedial from these neurons of ventromedial medulla, there will be release of GABA and glycine, which are inhibitory neurotransmitters, which will cause hyperpolarization and inhibition of brainstem motor neuron, thereby causing atonia. The second neurons which are responsible for this atonia of REM sleep are sublateral dorsal nucleus or a subcellulous area, which is a small cluster of cells ventral to locus cells. They release glutamate, which is an excitatory neurotransmitters, to the medullary reticular formations, which further release GABA and glycine, which are inhibitory neurotransmitters, and that will inhibit motor neurons and produce atonia in REMs. Now, there is a hypothesis, reciprocal interaction hypothesis, which was given by Hobson and McCarley. This explains how there is transition between RAM and NREM cycle. This is based on the interaction between the cholinergic and aminergic neuron in the mesopontine junction. So the cholinergic cells, which are also called as RAM1 cells, are in the lateral dorsal tegmental nuclei and pediculopontine tegmental nuclei. They are activated during REM sleep. And the serotonergic cells, which are called as RAM-off cells, are inhibitory to the RAM1 cells. These cells are most active during awakening and reduce activity during NREs. The REM sleep is terminated because the RAM-on cells are self-inhibitory and provide excitatory input to the RAM-off cells. So these are my references.
which is healthy for the brain. Describe the physiology. Medications, they decrease REM, increase REM, and that is useful in management of the disorder also. So what you give if there is like that's useful in the application part. So I think there is an application part of how you use that. The medication will be used. Night terrorist phase movement. So we differentiate terrors and BS. Night terrors and night terrors specific. Comes to can we differentiate the history that we get in the night and it's appearing here, which is again remember complete amnesia. Basically, substance the earlier part of the sleep so when REM starts, initial part of the sleep starts, then first REM comes. Conditions where it comes very early. In normal physiological conditions. Nowadays, the exam time chal raha hai. Eight. Follow the architecture. So what happens age-wise? What happens to MRE? Whether it decreases age-wise, REM decreases age-wise.
deep sleep or deep sleep being considered. Sure.
After consider you consider at least some exposure to right. Short chapter, छोटा chapter है तो that beginning part is also there.
No references.
That me है that me उसमें उसको करेगी
How much duration of sleep has in human life? Very, very important. What is the normal sleep duration? We're talking about physiology. What's the normal sleep duration? 8 hours. Someone is sleeping 8 hours and it's not. So what I see is explain very well. Nowadays, what I say on the more discussion side. Sleep physiology is a must. Sleep will in any phase in your exam. So, and it's one of the most common complaints. Okay, after a day, we get a complaint of sleeping. So, that is a very, very common aspect. And it's very complex. The more you go into sleep, it becomes more complex. It's not very, appears to be a very easy area. Right, most topics. So, chemical analysis or some lab measures are available. No lab measures.
just then imagine this
नॉर्मल स्लीप साइकिल कितना होता है कितने लेंथ ऑफ नॉर्मल स्लीप नॉर्मल ह्यूमन लेंथ ऑफ ह्यूमन स्लीप साइकिल फॉर ह्यूमन बीइंग्स नॉर्मल स्लीप साइकिल मतलब एक साइकिल पूरा स्लीप एक स्लीप एक साइकिल
मतलब 24 आवर्स होता है या ज्यादा होता है
नॉर्मली 4.5
So that is actually more than 24 hours. And then it resets after a few weeks. So actually, it's not exactly 24 hours. So human brain circadian rhythm just a little bit more than what this 24-hour cycle of desire is. So day-night cycle is 24 hours. But our brain is not switched. It's very beautifully in CGP. It's 25 hours. I was listening to what Naveen said that there is one disease where not allowed to sleep. If you sleep, you will vote for him. Huh? Next question. Next question.
Yeah. You have to move to respiratory, voluntary. So it is only voluntary. Like if you sleep, then you forget to breathe. That's the main thing. Okay.
Disorder with excessive sleep. What increase sleepiness. Hypo. Hyper. You said that you guys were going to go there. So that gets confusing.
Clearance,
It's a pushes. In anxiety, why we won't sleep? Or when we are distressed, we occupy. So why suddenly at night you don't get sleep? Mention
anxiety में क्या पड़ता है? You describe not of promoting wakefulness chemicals.
पड़ता है तो पड़ जाता है
Normally the anxiety and autonomic development होता है. और एक एक्टिविटी को डिस्टर्ब करता है. मैंने प्रॉब्लम होगा तो anxiety हो जाएगी. That is one of the mechanisms.
and then it leads to excellent pathway. Actually, then that is one.
She may not present because it is shorter part, but she should be ready for questions. Again, presentation part, maybe you can do it, but when you are on the dais, you should expect some questions, or the questions that you expect that should be covered. Covering, student, yeah, in 15 minutes you can only go this. Physiology books also. Brief mention is there.
mostly to treat the person about that.
I think that I missed the initial part of the presentation. I'm confident in it because the last presentation was given by one of your seniors. It was very tedious. The initial one was very tedious. So either a very good presentation or a very tedious, elaborate, exhaustive presentation. So, so this is a good attempt. Question answer, you should know the answers, you will have to be more confident. The voice volume should be more when you answer the questions. Okay, so that everyone knows from there. In terms, they might get in any of the discussions, they might get some answer right in that sense.
Presentation was okay. If you see the seminar, that part you covered there. Not much left to differentiate again. So that is why question is on applied part. But whenever you come, you should come prepared. These are basic questions that you face in. Okay, simple like you said, promoting wakefulness, promoting chemicals. So that's well pulling in one. So we should avoid giving cholinergic drug in the late in the night. So when you prescribe, I always say when you prescribe drugs even in TDS, so give the evening dose in 6, 7 p.m. It does not make sense to give tricyclics at bedtime. That because EPS is also not going to bother him in the night time and it will hamper the sleep. Okay, so sleep medicine is one different branch. It's coming up. You may end up having super specialization in that branch. So sleep, all of us need to know because as Dr. Robert said, insomnia is very common complaint. You see all the patients indoor, insomnia is a problem. And then you have to see for each patient, what is the problem? Whether he is getting some cholinergic or serotonergic drugs. Some patients, they develop insomnia secondary to SNRI or to SSRI, shifting them to morning. That may help. So you have to work for each patient. So you should know the basic physiology. Melatonin we are already using. We have other drugs that are acting on orexin that is available. Benzodiazepine. GABA, we can use it in insomnia also. So physiology part you should know. Second thing is that physiology you should know because polysomnography, you know, you have to grade different phases of sleep. It is important in diagnosing in which phase the problem is there. Okay, there is early onset of REM, so there are specific disorders. Okay. So this part you should. Depression, by the way, antidepressants decrease the REM, and in depression, the REM is decreased. And the SSRI will react. Third year. Oh, 30 years. 30 years, yeah, 5 years. 30 years, second year. 1 years only. 2 years only.
Asa is also big.
JRs are not supposed to comment on the quality of presentation. You have to speak on the topic.
After 4 years.
This is because you don't make a comment. Presidents are not supposed to.
Because this seminar is supposed, it appears that it is for consultant, the consultant. Faculty seminar.
Feedback is for improvement. You should give a comment.
So all the academics list is there, but all are supposed to come and lead. Whole house. Supposed to come and lead and then ask questions.
I mean, the city should be like. Yes,
So we'll also congratulate Dr. Singh. She is going to share the presentation. So I think a lot of thank
ठीक है। नहीं, लेकिन वो जो डिप्रेशन नहीं आता था।